So image analysis at the sort of highest level is a mixture of mathematical disciplines. And most of you are here generally because you probably don't like maths too much, right? A lot of you are microscopists, maybe biologists, physical scientists, and you want to use image analysis, but you don't really want to learn about the mathematics. Unfortunately, software is here, which can help utilize the power of these algorithms, of these mathematical disciplines, and present it to you in an intuitive way, which gives you an intuition for how a lot of these are working. But due to the way the GUI has been designed and the interactivity, it's much easier to use than potentially understanding the algorithm. But I do find that software is a great way of actually going on and understanding the maths as well, right? Once you have the intuition, then often the maths can follow as a consequence of that. So in the realm of microscopy, there's already a bewildering amount of software, and this is just one subset of all of the image analysis domains. So how do you navigate this space? How do you find what is appropriate for you? Well, you talk to people in the community that have some experience, and what I'm going to present to you today is one very important piece of image analysis software, which is primarily used for bioimage analysis, but can also be used for material sciences. And this is Fiji, and also it is known as ImageJ. So ImageJ was the original software, which was initiated in 1997 by Wayne Rasband. And then later, Fiji came along. So Fiji stands for, it doesn't really mean the country, but it stands for Fiji is just ImageJ. Okay, Fiji. So what the difference is that it has ImageJ at its heart, but it also has a lot of additional plugins and also infrastructure for getting to you the latest updates. So when you install Fiji, it also subscribes you to certain web servers, and these will allow you to update the plugins that you have and also download useful plugins which have been curated by the community. So don't bother downloading ImageJ, just download Fiji, because it's essentially the same thing, but it has more functionality which is useful for you. Okay, so you can download it here, or just search for Fiji and download. You won't download the country, it'll go straight to the software. So I'm going to introduce to you a little bit about image representation. So an image is displayed to you visually as a picture, but underlying that image is a 2D array of numbers. In the case of an image like this, there is one number for each pixel. For a color image, there'd be more than one. Okay. In terms of the monitor, it will display that image as red, green, and blue. Okay. But for a grayscale image and a lot of microscopy images, you would just have one pixel, one value per pixel in your image. And because underneath it's just an array of numbers, then you can do statistics and other measurements on your image. So, for example, we can take the min of this 2D array. We can find the minimum value. We can find the maximum value. We can measure the mean, the median, the standard deviation. We can apply all kinds of statistics because it's just an array of numbers, a list of numbers. For example, here we can draw an arbitrary line across our image, and we can sample the underlying array and then present it like a line profile, like we've got here on the right-hand side, where the higher values represent more intense values. Okay. And in the case of this image, a bright pixel is a larger number. So, for example, white is 255 and black would be 0. But you can flip those values round if it suits you. And then an image is quantized spatially. So what I mean by this is there's an origin, and often in image analysis the origin is in the top left-hand corner, which is different to, say, a graph where it's in the bottom left-hand corner. And also there is a resolution associated with your image. And in the context of image analysis, this represents how many pixels that compose your image. So how many across in terms of its width, the number of columns, and the number of rows. So how many pixels compose the height of your image. And the more pixels you have represents a higher resolution. Now this is somewhat distinct from microscopy resolution, where we use resolution to describe the resolving power of your microscope, so the optical ability to resolve certain details. So we use the same word, but it has a somewhat different meaning. But when you do an imaging experiment, it's not always the best idea to have the highest resolution possible. You should really potentially downsample your image in terms of resolution appropriately to the question that you're trying to ask. So for example, you may have a microscope that's resolving individual organelles, but the question you're asking is really about how many cells do I have in an image as part of a population. And in that case, you could actually lower the resolution so that you can just resolve the individual cells rather than necessarily being able to resolve the organelles. And the reason you would do that, the reason you would throw away that data, is so that you could save on memory and also on processing speed. So a larger image, and this is a theme that will come up again, a larger image is slower and more expensive to process. So if you can, and if it doesn't contravene any of your other obligations, it's always a good idea to downsample with the resolution if you can. So in the way that your image is quantized spatially, it's also quantized in terms of intensity. So bit is terminology used in computing to describe memory, and you'll often see that an image has a certain bit depth associated with it. So this is a kind of fictional bit depth for 3-bit, and this represents 2 to the power of 3 levels, so 8 levels. And what this means is that this image here, which is the same image as before, is now described with 8 levels between the brightest and the darkest shade. Okay, and you can barely make out what the image is of because that's not very many. And computers have moved on a long way since we were limited this dramatically in terms of memory. Okay, so 5-bit is a bit closer to where we are now, and you can see there are now many more levels, many more shades between the brightest shade and the darkest shade. This is a 5-bit image, which is 2 to the 5-bit, 32 levels, and you can see that the apple is now much easier to see against the background. But what is very common is 8-bit images and also 16-bit images, and you might have seen 12-bit images and even 10-bit images. So here we have many more levels of grey. So an 8-bit image is 256 levels going between the brightest shade and the darkest shade, and you can't actually see the steps anymore. It's a smooth continuum. And the reason I've got 8-bit and 16-bit in the same area there is because most conventional monitors and projectors can only display subtleties up to 8-bit. So although the information exists at a higher bit depth, the monitor that you're using can't actually show you those subtleties. And that we as humans have limitations as well. But in terms of the underlying data, you can use that information. And now our detectors on our microscopes are often capable of holding bit depths of more levels than 8-bit. So we can acquire data at that level and we can analyze it. But visually, you won't necessarily see a difference unless you do something to manipulate that range to focus in on a certain sub-range, for example. Now, there are TVs and things now which can do 10-bit. I believe HDR TVs are 10-bit, which represents a new sort of evolution of data format, and it will require more memory, obviously, to store that and to distribute it around around the world. But in terms of most conventional monitors and projectors, 8-bit will be their limit for a while until more technology comes along. So just be aware that although you can't see the difference, you might want a superior bit depth to 8-bit. You might even want more in terms of your analysis. So it'll allow you potentially to analyze subtle details in your image. So be aware of that. And if you don't need it, for example, if you're just counting very coarse features, then you could also downsample to 8-bit and save on memory and processing time as well. Okay. So when you open an image in Fiji ImageJ, and I'll show you this later, it will appear and you'll have the graphical representation. But when you move your mouse over it, you'll see the coordinates of where your mouse is relative to the basis of the image. And you'll also have a value which represents the intensity. So this is this value between, say, 0 and 255 or 0 and 65,535. And if you move it around, you'll see that value changing. Now, in this image, the value is actually a floating point number. It has a decimal point. And this represents the cursor being on a pixel, but rather than having an integer number like we had with 8-bit and 16-bit, we have a decimal point now. And if you see here, it has a bit depth of 32 bits. Okay, so this isn't just a larger number than 16-bit. It actually represents 2 numbers, the number before the decimal point, the number after the decimal point, and what you don't see is the position of the decimal point. as in there's some memory allocated to where that decimal point appears. So what 32-bit depth means is that we can have floating point numbers. Okay. Before we had integer numbers and a different amount of them, but 32-bit specifically in this case, and also you'll find 64-bit float in other areas of image analysis, refers to floating point numbers that have a decimal point. Now, why would you need that? If an integer was fine before, why do we need floating point now? Well, the answer is when you do some mathematical operations on an image, it may be very beneficial to have a floating point there. So if our analysis involved dividing this image by 2, like a simple mathematical operation, then we would have 0.5s in our image. Every odd pixel value that we had before as an integer would become a 0.5. And if you just have a conventional bit depth, an 8-bit or a 16-bit, then you wouldn't be able to have 0.5. It would be rounded to the nearest integer. So if you were then to multiply that image by another number for another operation, you would lose information because you no longer had that 0.5, which would be multiplied to its own discrete integer. It would be lost in the rounding. So we use floating point numbers to ensure that we are consistent mathematically if we're doing lots of kind of mathematical operations on the image, which can be a natural consequence of doing image analysis. So the caveat of using 32-bit is, again, it requires more memory than a 16-bit image. It requires more memory than an 8-bit image. So really, you should only use it when you need it. Okay, but just know it exists and that it's for floating point numbers. Okay, and now this is probably the most important part of the talk. So when you've got images on a computer, the underlying array, which I've talked about before, that information is sent to your graphics card, which then decides what color to present it to the screen. Now, I've shown you the grayscale image, and this means that an array with a single value for each pixel is telling the graphics card that we want red, green, and blue all to show the same value. That's what a grayscale image is. It's when you have an RGB display and it's telling each color to be the same level. That's why it appears grey. Maybe a bit counterintuitive, but it's composed of red, green, and blue. If we have a red image here, we're basically saying we only want the red pixels in the display to show the value and the other ones are off. And conversely for blue and for green. So what happens is there's an array there, a data array, and in the case of Fiji ImageJ, the color is somewhat independent of that. As in, you can color it any way you like and the underlying data stays the same, and it's up to you as a user in which color that appears. So for example, a microscope will have a detector and it just counts photons. The detector isn't aware of what color those photons are, what wavelength they are. It just counts them. It's then up to the software to color that array, and the way that will be done will be in the metadata of your image file. So this is the data, the information which accompanies your raw pixel data. So sometimes you can save fluorescence that's been caught within a certain range due to the filters, and the detector just counts the photons, and then the software will say, it will store in the image file what filter was there, so what range those pixels were from, and it will assign it an arbitrary color. But that color can be changed very easily. You just choose a different lookup table. And so if you see a blue image here, it doesn't necessarily mean it was acquired within the blue range of light. It could have been the green range of light, but the software, through your changing it, or through a mistake in the way that the software has been designed, it might have given it a different color. So don't trust the color of an image, especially in image analysis software. Know that underneath it there's data, and that the color is an extra assignment put on top of that. But we call these assignment tables lookup tables. So it takes the intensity and it looks up basically how to display that to the monitor. So these are quite conventional ones. So this is a grayscale one, this is a red lookup table, a green lookup table, blue, cyan, magenta, and then we've got a bit of a special one here where we're not just mapping to shades of a color, we're mapping to different colors. And so this is the same underlying data, it's just one value per pixel, but we've said when it's particularly bright we want it to be red or white, and then when it gets more dim it's blue or black. And you can interchange between these very easily when it's still in the image analysis software. Okay. And these ones are good, for example, if you want to show a subtle range more discreetly. So we're actually less sensitive to blue than we are to green and red. And so you might really want to use a kind of bluish one, but you could use these colors, these more elaborate lookup tables to map to colors which you can more easily identify within certain ranges. So it's a way of expressing the data in certain areas more vibrantly. Now, image analysis has the interesting property that, or microscopy has the interesting property that you can acquire multiple channels in a single image. So this is a case of a hippocampal neuron which has been stained in multiple colors and also imaged in two different modalities. We have fluorescence modality and we have a bright field modality. And what's good is that we can actually store all of this information in the same file as separate channels. And a conventional way of doing this would be to use the microscopy's own format, or we can use a Tiff format. So Tiff is independent of any one microscopy company, and actually it comes from the days when we were using faxes. So it's a multi-page format, so each file can store multiple pages, and that comes from when you were doing faxes and you were doing multiple pages. So the microscopy people hijacked that, and rather than having multiple pages of writing, they have multiple pages of, well, multiple channels of information, which represent different detectors and different acquisition properties on your microscope. Now, just to point this out, this isn't a channel in itself, this is a composite representation of all of these channels, whereby we're basically overlaying them and weighting the pixel colors equally, and that just allows us to see the whole image in one go. But if you keep your image stored in the proprietary format of the microscope or in a TIFF, then you can save the data and then open it, and your channels will still be independent. Now, this is discrete from photography formats. So, for example, if you saved a 5-channel image as a JPEG, this is a photography format, so it just has red, green, and blue underneath it. So it would look the same as this. It would look like a composite representation, but when you came to split the image again, you would only have 3 channels, and your information has been corrupted between those channels. It still looks the same when you merge it, but it's not storing it independently. So when you do image analysis and you're storing data for data analysis, you have to be very careful about what format you use. Don't use photography formats like JPEG, PNG, or GIF, for example. Now, those formats have their uses. For example, they are very efficient, and if you're making figures, then you can use them because you're just visually showing your data. As long as you don't use compression and things like that, you can still use JPEG. But just so you're aware, it's not good before data analysis. And actually in our institute, someone on their microscope actually saved out their image as a JPEG. This was an early microscope before they kind of learnt their lesson. But they had 3 lookup tables, which were green, cyan, and red. Now, if there had been red, green, and blue lookup tables for 3 independent channels, then they would have split quite nicely. But because they had a cyan lookup table, when they passed their image to the image analyst, he split the image into red, green, and blue because it just had a JPEG. And actually, the 2 of the channels, the green and the blue, were subtly contaminated with one another because when it was split, it didn't know what the actual lookup tables were, so the information had been destroyed or corrupted in that process. And it got all the way to publication. And then I realized that their data had been on some level corrupted. Fortunately, it didn't change the conclusions of the paper, but that was kind of lucky because the trends were strong enough to override this contamination between the 2. But yeah, it's not good. So really be careful about this. So now we don't have loads of time, so I'm rushing through a bit, but I'm going to take you now through some typical analysis to give you an idea of some of the terms that come up. And segmentation is probably one of the most common ones. So imagine this is our input image. What we want to do is we want to threshold this image so that we can express it more in line with what a computer is used to doing. And I'll show you why that's important. But the way in which we do segmentation is that we display our image like a histogram, first of all. And a histogram in this context is that rather than showing our information spatially, what we're doing is we're counting our pixels of each intensity and then displaying it like a plot like this. So this peak here represents our most frequent pixel, which isn't black, it's actually an off shade of blue. So our most common pixel is our background, and it's around this intensity here, which is probably like 25 or something. And then as we go brighter and brighter, this represents the intensities of our cell. But if you saw this, you'd you'd generally think this was our cells, right? When you look at this image, you think that actually the most common pixel is from our cells. It's from our cells rather than the background. Maybe I'm wrong, but actually this is our background and it's not black, but our eyes are quite bad at determining that. So a histogram is useful for inspecting our image. And what we do when we do segmentation is that we're basically trying to pick a value on this scale which will separate our image nicely between foreground and background. So like this, for example. So we have now represented our background pixels as white and our foreground pixels as black. And now just how I've described it is that we've just chosen a level. But as part of a macro script or as part of a pipeline, you'd often not want to make that decision yourself because that introduces your human subjectivity. You'd want to use an algorithm, for example, the UTSU algorithm. So what that does is it interrogates this histogram and it picks a level which it thinks it's appropriate, and then you'll have the result of the foreground and the background separated. So this image at the end is what we call a binary image. It's sort of on and off. Okay, you're either background or foreground. And once we're in this form, we can start doing things. But this is the basic pipeline for segmentation. Either you'll choose the threshold yourself, which is it's okay if you're choosing a fixed value and you keep to that value, or if you're going to use a dynamic algorithm which can be more resistant to effects like noise and things like that, and it's not subjective, it's less subjective than you. If you have to keep changing it for your different images, then I'd recommend using an algorithm like UTSU, or there are many other algorithms like max entropy and Huang, which you can choose from in the image Fiji threshold setting, which I'll show you in the practical part. But once your image is in a binary form, then there are many things that you can do. This is very amenable to kind of morphological operators, which were like the hallmark of image analysis in the recent decades. So, for example, we could find the outline of our spots here, which could be used, for example, later on for measuring the intensity of a membrane stain or something like that. Or we could use a watershed operation to split our bigger blobs and try and find the individual cells for counting or measuring, taking measurements for each cell. Or we could erode our blobs and potentially just focus on the nucleus, or enlarge them to try and find something that's outside the cells. So there are many ways in which you can use combinations of these operators to do different kinds of analysis. And this is very typical in conventional signal processing, that we will segment or threshold our image, and then we will do things with that mask that we get, and then apply it to make measurements potentially on our original image. So one of the important things, once you've thresholded your image and you've got the binary representation, is the parameterization. How do we get the locations of the cells? How do we get areas unique to each one? Because before we did this, we just had black and white pixels, but now we have a numbered area for each pixel, so we can actually isolate each of the cells individually and take measurements with each of those individual masks. And for that, we use the Analyze Particles plugin, and then it puts in the ROI manager, region of interest manager, an entry for every cell, so you can select each one and independently manipulate them. So this is what I describe as parameterizing the objects in your scene. And I've shown you segmentation. That's one way of finding this parameterization. Another really useful algorithm, which is very simple to use, is the Phi Maximum plugin. So in my institute, they do a lot of foci analysis on nuclei. And this basically means a lot of counting bright blobs, And sometimes when you use segmentation, because it's a global threshold, it applies the same threshold across the image. If you have sort of background effects which have an intensity profile like this, a threshold in the middle of the image isn't always appropriate on the edge. So an algorithm like this, a Phi Maximum algorithm, is great because this looks for local fluorescence maxima. So it looks for bright blobs in a certain area. So a bright blob on the edge of the screen may not have the same maxima as one in the middle, but because it's locally dominant, it still gets picked out. So Phi Maximum is great if your background's very variable, but you as a human can see bright blobs. So it has one parameter, which is the noise tolerance. And what this does is, when you have a very low noise tolerance, it will find many maxima. And then as you increase the noise tolerance, it will just find the more dominant maxima. So there's a certain protocol behind this which looks at the nearest peaks and decides if they're within the threshold or not, and then it will prune them or it will merge them based on how similar the values are. So if you have a very high noise tolerance, it will find very few maxima. And what you would do typically for an experiment is you take several test images and you would find a noise tolerance which kind of works for all of them, and then you would apply the same algorithm to each image and then use those locations either to take measurements, to count them, or to be as part of some kind of deeper analysis. Perhaps you're looking at a distribution or measuring the distance between them. So then I just want to talk about macro writing. So I actually got into computer science. I used to program games when I was young, but I didn't think there was a future in computer science. So when I went to university, I did cell biology, and then I did a PhD in neuroscience, which involved a lot of imaging. And I knew I could kind of program deep down, but I wasn't that happy with how to do it. I was trying to do things that were too complicated, really. But I found the visual aspect of Fiji ImageJ really nice. So if you find the record functionality, you can actually record the menu items that you're clicking. So the threshold tools and the analyze particles, when you use them just as a user, they'll appear in the record function. And then it's quite simple to make scripts out of this record function, which allow you to repeat your analysis the same way every time. So for me, because I'm quite a visual person, it was nice to be able to do the steps with the software and then see how they've been described with the functions. And then it used to be a bit harder, but now there are loads of good resources online. And I talk about it later, but you can basically ask any question you want online, and there'll be someone now who can help you do it with the scripting. So it's never been a better time to learn to script. And for me, this was a great way of actually getting in. And now I program in all types of languages, and you wouldn't know my origins, basically. It's written in Java originally, but the scripting language can be many different languages. So they have something called Jython, if you like Python, which is like using the same syntax as Python, but it speaks to Java. They also have JavaScript. They also have ImageJ1 macro language, which is their own one, and they also have the Groovy, which is getting very popular. So if you can program any of those, you can use the scripting language. If you want to develop plugins, that's still in Java. So metadata is very important. I mentioned that your microscope will embed metadata in the image format, and with ImageJ Fiji you can easily look at that. So you just open it in the menu under image and then show info, and then you display all the settings of your microscope and things like that. So if I suspect that someone's giving me data that hasn't been acquired in a reproducible way, I go to the metadata and I see exactly what settings they used on the microscope. So there's really useful for keeping a track of how that image was acquired. And in ImageJ Fiji, you can access that very readily. And then, so that's the end of the slide-based bits. I'm going to do a practical demonstration now. But I just want to point out to you that I'm actually here. The reason I got my VIP lanyard isn't for doing the learning zone. It's because I'm part of the Image Analysis Focused Interest Group of the RMS. And this is a group which have a kind of the remit of bringing together a community of bioimage analysts. So this isn't just people that are real image analysts from a physics background or something like that. This is people, biologists and microscopists and physical scientists that use some kind of image analysis in their work, and they want to be tapped into the community in terms of accessing resources and gaining support should they want to proceed in that particular career structure. So I'd really encourage you to take a note of the website, and also preferably also is to join the mailing list, or you can come and talk to me afterwards and I'll add you to the mailing list. And this just updates you with things like training events and committee meetings and things like that, which you can join in on. But if you have, this isn't really a list for asking questions about image analysis. What I would really recommend for that is the image.sc forum, because a lot of the people doing image analysis in this community, and also New Bias, which is like the EU-wide sort of umbrella for image analysis, a lot of their members are here, and this is really the place to be asking questions about how to do things. And it's been taken on by many of the big software people for bioimage analysis. So really, this is where everyone converges, and it's a really nice forum. People are very responsive, and a lot of people are using it. So this is really good. And also there's a New Bias conference, so this is what I mentioned, this is the finale unfortunately, but this is a really great conference to learn about image analysis and to interact with experts and other users. It's a really sort of warm and nice conference to be involved in. So that's in Bordeaux in March. But now I'll